Well, good afternoon, everybody, and welcome to our last Dean's Lecture of the year. We've had a great series, and it's been a real pleasure to introduce some of the most renowned of our faculty who have been recently promoted or recruited into the position as professor here at the School of Public Health. And this week, Dean's Lecture is no exception. We are here to honor and learn from Dr. Daniela Drummond-Barbosa, Professor of Biochemistry and Molecular Biology. One of the fun parts about being Dean I've learned over the last several months is to get to meet people I would not normally meet as a professor in health policy and management, and to learn new areas that are very much outside my own area of expertise. And I really enjoyed, I spent a few hours with the Department of Biochemistry earlier in the year and had a lot of fun just learning about what's being done. And they even brought me into the lab and had me pipetting, and it was great fun, so I appreciate the time you spent with me. Today we're going to hear from Dr. Drummond-Barbosa. And she pioneered the use of Drosophila to study adult stem cell regulation by diet. She showed that the ovarian stem cells and their descendants proliferate and grow faster on rich relative to poor diets. Her laboratory subsequently played a major role in delineating how insulin-like peptides, ectozyme, and the target of rapamycin nutrient sensor mediate this response by acting in the ovary. And more recently, her program has been addressing the link between adipocyte physiology and stem cell biology. And this question is particularly relevant to our understanding of the current obesity epidemic and the poorly understood relationship between obesity and cancer. Before coming to the Bloomberg School in 2009 as an assistant professor in BMB, Daniela was an assistant professor in the Department of Cell and Developmental Biology at Vanderbilt University Medical Center. She was promoted to professor in 2016 and holds a joint appointment in our Department of Environmental Health and Engineering. She is currently principal investigator on 3 R01s from the National Institute of General Medical Sciences, and she is a terrific advisor to many students and postdoctoral students and instructs the course Stem Cells and the Biology of Aging and Disease. Daniela is the recipient of numerous honors and awards, including being elected as a Fellow of the American Association for the Advancement of Science, the Shikan El-Hibri Prize for Discovery and Innovation, the Research Scholar Award from the American Cancer Society, and the Chancellor's Award for Research from Vanderbilt University. Daniela received her PhD in genetics at Yale University School of Medicine, and she completed two postdoctoral fellowships, one at Yale under Dr. Danielle DeMaio and one at Carnegie with Dr. Ellen Spradlin. Please join me in welcoming Professor Daniela Drummond-Barbosa.
Well, thank you for this very nice introduction, and thank you for the opportunity to share my research with the whole school and beyond the school, across the street too. I'll start by introducing the problem or the question that my lab has been interested in since I first started it at Vanderbilt. And the question is, how stem cells in multicellular organisms that have very complex physiology and can respond to lots of different changing situations, how do they sense and respond to changes in the physiology of the organism as a whole, which can happen in response to stress, diet, aging, exercise, and a number of diseases. And as you can imagine, this is a fundamental question in biology. But it's also, as the dean mentioned, it's not too hard to imagine what the potential public health relevance of studying this problem could be. But before I talk about the problem that my lab is interested in, I want to make the point that when it comes to basic research, oftentimes when the questions are being asked about a fundamental problem, you cannot imagine what the public health relevance of that question is. And only after many years of research is done that you can see that there was a tremendous impact. So the point that I would like to make is to defend the idea, which is my view of the role that basic science could play in a school of public health, making the case for curiosity-driven basic research. So even if you cannot necessarily articulate precisely what the public health relevance would be. And there is many, many different examples of questions which at the time they were asked, people had no idea what they had to do with anything that anyone should care about. But as years went by, they had the hugest impact on public health and well-being. And honestly, we could talk all day long for many, many months describing all these examples, but I think you probably want me to talk about my research a little bit too, so I'm just gonna focus on 3 that happen to be my personal favorites, but people could have picked anything they wanted. So one of my favorite weird questions that were asked many years ago is, how do bacteria destroy foreign DNA that they don't want? It's kind of hard to imagine how that question could have much of an impact. But it turns out at that time, it led to the discovery of restriction enzymes. And as everyone in the biomedical lab sciences know, these restriction enzymes are incredibly useful for cutting and pasting DNA together, and it allowed the molecular biology revolution, which basically changed the way everybody did research and allowed everybody to really start investigating all kinds of mechanisms. So that's one example I really like. Another example, which is a question that occurred to someone, what are these weird repetitive sequences doing in this bacterial genome? And so that's also you see some repetitive sequence, you think, oh, this is just not worth my time, and it would be really hard to justify to the NIH studying some random weird repetitive sequences in DNA. But it turns out, through a convergence of other studies and many studies later, that this actually had everything to do with the CRISPR systems that bacteria have, which also kind of help answer this other question here. And one of these CRISPR systems actually was adapted for technological purposes and is leading now to this gene editing revolution. And not just gene editing, but a ton of other applications where you need to just target things to a specific region of the genome. So that's something else that you could never have predicted. Now, my very favorite question is, not that I'm biased or anything, it's how do fruit fly newly fertilized eggs, which don't have any apparent pattern to them, how do they become segmented larvae? That one is really hard to imagine who should care about that question, but except people who love fruit flies like me. But it turns out, at the time this research was done, there were a lot of novel signaling pathways that were discovered for the first time in any organism, in fruit flies. And it turns out, yeah, all the signaling pathways were involved in segmenting the larvae. But people later wondered, are there homologs of these genes in mammals? And it turns out they were, and they play all kinds of incredibly important roles in development and disease. They are conserved across all multicellular organisms, and they are used over and over and over again because there's only so many pathways, so you have to keep using the same ones to do everything you need to do if you're a living organism. So these are just 3 examples that I like, but there's lots more. So my point is that, of course, we do need, if you think about a retirement investment, the closer you are to retirement, you need to have more safer short-term investments. But if you're far away from retirement, you also have to think of investments where you don't really know how they're going to do, but they are kind of high risk, high rewards. So it's kind of similar when it comes to the role of research in a visionary zoo of public health, where you should really have representation from all parts of this portfolio, that we do need to have a lot of research where you can see the impact in the next 5 years and change laws and improve screening ways and figuring out ways to change the way people behave for the better. But if you really want to have a vision that goes 50, 100 years into the future where we're going to be having an impact that far, you really need to have strong support for basic research. And as long as a fundamental, interesting problem that's going to matter to a living organism is being investigated, it's more or less guaranteed that there will be some relevance, because we all evolved from a common organism when whatever is really crucial for an organism to function is going to be conserved. So now, going back to my research, which probably falls somewhere along in the middle of this continuum here. So how did we go about studying how stem cells are controlled by physiology? As I already hinted at based on my favorite question, we use Drosophila melanogaster, fruit flies, which, as you can see, is a beautiful model organism. But it's also very, very useful, and it's much bigger than this in all our hearts, like in the lab. And so in Drosophila, sorry, this might be a little small. There is evolutionary conservation of basic cellular molecular mechanisms. You can take advantage of genetics, cell biology, physiology, and it's for the purpose of studying physiology, where you're thinking about lots of different tissues and lots of different cells in an organism. One particular advantage of Drosophila is that it's highly amenable to cell and tissue-specific manipulations, so you can disturb genes and proteins in particular parts of the flies and see what function it plays in that part versus another part of the organism. And no wonder this organism has been the model organism that led to a lot of discoveries that were worthy of the Nobel Prize. And these are some that I had from an old slide, but last year I had to add one more for the research that was done on the circadian clocks that was in Drosophila. And this is actually the research, these two scientists did the research on how the larva becomes segmented. And this person here discovered all the Hox genes that conserve all the way from flies to humans and that control the identity of different body parts along the AP, anterior-posterior axis. So I hope you all think that Drosophila is a very worthwhile organism to use in research by now. Otherwise, you can just not listen to the rest of the talk. But, and just to give you a little bit more information about how Drosophila relates to humans, of course, Drosophila doesn't have all the tissues, all the organs that humans have, like as specialized as they are. But ultimately, as a multicellular organism, Drosophila has to fulfill all the functions, pretty much, that are really important for any multicellular organism. So they have, for example, adipocytes that correspond to the adipose tissue. They have cells that are like hepatocytes, and they have basically some cells that will serve the purpose of all the organs. And they also have very similar nutrient sensing pathways and other physiological pathways that help all the organs communicate, for example, the nutritional status of organisms. And of particular interest here are the adipocytes, as you see later in the talk, that happens to be a particular area of focus of my lab. So I introduced you to Drosophila, and now I just want to introduce you to the particular organ in Drosophila that we use to study stem cells, and these are the fruit fly ovaries. They are quite large. This is a pair of ovaries. They take up most of the abdomen of the fly. And you can see at the anterior end here is where the stem cells are located. And each of these ovaries is composed of a lot of different ovarioles. And as the as the eggs develop in the ovarioles, they are laid by the fly through the posterior. And if you look at one of these ovarioles, this is, of course, a cartoon, not the real one, but you can see there are progressively older follicles, so like beads on a string. And these follicles or egg chambers have, you can imagine this is like a watermelon, except the colors are inverted, but in the middle, you have the germline cyst, which has 16 germ cells. One of them is the oocytes. The other 15 are nerve cells, which just support the growth of the oocyte. And outside, surrounding all this germline cyst, are follicle cells. And this, unlike in women, where early stages of germ cells happen during while they're still embryos, and the other stages happen during adult stages, in Drosophila there is a timing difference in where there is a constant supply of precursor cells here that make up eggs throughout the life of an adult fly. And these are inside the germarium. So if you look at a higher magnification of the germarium, you see that there are 2 germline stem cells here that give rise eventually to this germline cysts here. And then eventually they become enveloped by the follicle cells. And those germline stem cells reside in a special location or niche. And the cell type, the major cell type in the niche is called the cap cells here in yellow. And this specialized niche environment gives the stem cells special signals and in order to maintain them as stem cells. But like I said, this is just a diagram, and just to give you an idea of what these things look like when you look at a confocal microscope. You can see here, this is one stem cell here, this is another stem cell, and in green here, you see a niche of those cap cells, and in pink here is a kind of molecular glue made up of this molecule called E-cadherin that helps, like Velcro, the germline stem cells get stuck to the niche so they can be exposed to that specialized environment. So another a characteristic of the ovary that was very interesting when I first started my postdoc, and I thought I could exploit that to understand the regulation of stem cells in response to physiology, is that the ovary responds very drastically to diet. So this is a graph showing the number of eggs per female. So the females eclose, and then if you keep them on a rich diet, which is basically providing a lot of wet yeast paste, the yeast that you use to bake bread or make beer. So they love that. So they'll lay lots of eggs, so each female can lay up to 80 eggs per day, which is a lot considering the size of the egg. But if you take all the yeast away from the diet, they'll stop laying eggs almost immediately. And conversely, if you put this newly eclosed females that just came out of the pupal case on a poor diet, they won't lay very many eggs. And then as soon as you switch them to a rich diet, they lay lots of eggs. And if you dissect the ovaries, for example, this ovary was dissected at this time point, you can see on a rich diet, you have very nice ovaries with all stages that are well represented. If the females never ate yeast, the ovaries look tiny. And if they were eating yeast and were switched to a poor diet, you can see that on the surface it looks like the ovary is still big, but these are all the last stage of oogenesis. where the ovaries are just being held, the eggs are being held inside the egg, the female, waiting for a better opportunity to be laid. And there's a lot of intermediate stages of oogenesis missing here. And this is summarized in this slide here, where, as I mentioned, this last stage is controlled so that if there is no yeast in the diet, they delay ovulation. And all the vitellogenic stages where the oocyte has started taking up yolk and growing a lot are also missing, because these stages here will degenerate if there is not enough yeast on the diet. And these stages are still there. But when I started my postdoc and started asking this question, we didn't know if there was any regulation at these early stages in response to diet. And what I showed during my postdoc, which served as a foundation to start my lab, was that there was regulation of how fast the stem cells can generate daughter cells that will give rise to new eggs, how fast the daughters of the stem cells can develop and how well they survive. And early on when I started my lab, I also found out that the number of stem cells in the niche was regulated by diet, and all that together led to this regulation in egg production. So the question that I've been asking since the beginning of starting my lab is, what are the physiological mechanisms mediating this response to diet? And because this is a broad audience, and the point is not to give an exhaustive account of what we've been working on and bore everyone to death, I decided to just highlight two different aspects of our findings, which I think illustrate the kinds of questions we ask and also the kinds of approaches that we use. One is I'll focus on our investigation on the role of insulin signaling in directly controlling the germline stem cell lineage. And in the second part, I'll talk a little bit more on what the dean mentioned during the introduction, which is the adipocyte-ovary communication, of how adipocyte can send messages to the ovary to modulate the stem cell lineage. So starting with the first question, first I'll just give you a very brief introduction of the insulin signaling pathway. In mammals, insulin is made in the pancreas. In Drosophila and other invertebrates, is made in neuroendocrine cells, but in both cases, these are regulated by diet. The insulin that's secreted act on receptors at the membrane, which then act through a cascade of downstream effectors that control a lot of different aspects of metabolism, physiology, growth. And of course, if something goes wrong with this pathway, you have a lot of different pathology that can result from that. So doing our research, we manipulate the insulin receptor if we want to vary the activity of the pathway, and we also manipulate other components of the insulin signaling pathway to change its function either in the whole organism or in specific cells. So one observation early on during our research, as I already mentioned, is that we measured how fast the stem cells, the germline stem cells, were proliferating or dividing to give rise to new daughters that can then make more oocytes. And we used this marker at the time called BRDU, which basically gets incorporated into the DNA of dividing cells, so that you essentially can take a snapshot of how often cells are dividing in a population. So if you see more cells, they're dividing faster. If you see fewer cells, they're dividing more slowly. So as I said in that introductory slide, on a rich versus poor diet, there is a difference in the proliferation rate, the division rate of those germline stem cells. And when we looked at two different mutations in the insulin signaling pathway, we saw that there was also a decrease in proliferation, suggesting that the insulin pathway is one of the pathways that gets regulated by diet and mediate this effect of diet. But in this case here, the entire fly was mutant for the insulin pathway. So every cell in the body of the fly was mutant. So that doesn't allow you to tell whether the insulin signals are sent directly to the germline stem cell to directly instruct the stem cell to divide differently, or if there is some intermediate step in the body through another cell type. So the way you can address this question in flies, where this is very easy, I won't go through the particular details here, but the point is that you start with an organism where all cells have a wild type, at least one wild type copy of the insulin receptor, so all cells can respond to insulin. And then you use genetic tools in flies, and you create subsets of cells that happen to be mutant for the receptor. So using this kind of approach, you can get rid of the receptor either in the germline stem cells or in other cells and find out if the germline stem cells themselves need the receptor in order to respond to changes in insulin signaling. So, just to recap, like when a germline stem cell divides, it forms a daughter cell, which is called the cystoblast, and that will divide to make up a 16-cell cyst. So if you look inside the germarium, which is where the germline stem cells are, each product that you see, one of these cysts, corresponds to one time in the recent past that the germline stem cell divided. So if you have, for example, in this germarium here, you can have the red cells are cells that have a wild type insulin receptor, so they can respond to insulin just fine. These black cells here do not have any good copy of the insulin receptor, so this cell is defective for responding to insulin. So if we count how many of these products we can find for a wild type cell inside the germarium and how many you can find for the mutant cell, you can essentially tell how many times each of these germline stem cells divided in recent past. So if they divided the same number of times, the ratio of number of their progeny would be 1, because there would be equal numbers of both descendants of both stem cells. And that's what we see for the control. But when we looked at 3 different kinds of mutations in the insulin receptor, every single time the germline stem cell was mutant for this receptor, it could not divide at the same rate as the wild type. So this really showed that the insulin-like peptides directly tell the stem cells to divide at different rates. So this just illustrates how we figure out the physiology of the signaling pathways. And it's not, we also looked beyond the germline stem cells, and just to illustrate, this is a control where all cells are wild type, and as you see that progressively older follicles. Now again, if we look here, the red cells have wild type insulin receptor, the black ones have mutant. And as you can see, these 2 cysts here that are mutant for the insulin receptor, they can't grow properly, and you can tell they're too small compared to the neighboring cysts. So this also showed then that insulin signaling directly controls how fast the follicle grows. And that's also true if you mutate this other pathway called the TOR pathway, which will come up later again in the talk. But this pathway senses a lot of different kinds of inputs, including the levels of insulin signaling. And in this case, these 2 pathways work together to control the cyst growth. And interesting, if you mutate a negative regulator of this pathway, the cyst will grow too much instead. And in all these cases, the pathways were mutated in the germline. But we showed that even if we mutate the pathway in the follicle cells, sorry to switch the colors. Here, the greens are wild type and the blacks are mutant. So here, like if the mutations are just in the follicle cells, if this is a mutation in this same pathway in the follicle cells, the underlying wild type cyst will also grow more slowly. And if you do this negative regulator mutation so that the pathway is overactive in the follicle cells, you also see overgrowth. So this tells not just that these pathways control the growth, but that they can control from either the follicle cells or the germline, and that there is extensive communication between the two cell types. So this is just to illustrate. And interesting enough, like this is very similar regulation of the mammalian ovary is also happening at the level of the growth of the follicle. Because if you delete Pten, which is a negative regulator of the insulin TOR signaling in the oocyte, the mammalian oocyte, because normally each month a few of the follicles will grow, and then you ovulate. The ones that don't get ovulated die, but you still have a reserve of primordial follicles. Now, if you mutate this, of course this is done in mice, not people, because you can't do that. But so if you mutate this pathway in the germline of mice, there will be so much growth too soon of all the oocytes that it will deplete the pool, and the females will stop producing new pups compared to the controls. So just to summarize then to this point, in the initial phase of my lab, we were focusing a lot of how insulin-like peptides control these various aspects that I just mentioned to you, and also vitellogenesis, the yolk uptake. We also studied a lot of other nutrient-dependent pathways that I don't really have too much time to talk about, all of them acting within the ovary. But of course, we know that all organs are constantly talking to each other, because there's all the multicellular organism doesn't have a bunch of like independent islands in them. They are all communicating with each other. So the question is, of course, a lot more complex, where you have not just the brain that makes insulin-like peptides in the ovary, but you have gut, adipocytes, muscle, and lots of other tissues, and they're all communicating in all kinds of directions. And ideally, it would be nice to know all that, but it's not going to happen in any of our lifetimes and our children's lifetimes and grandchildren's lifetimes. But so instead, we decided to focus on one of those multiple arrows at a time, and that's the arrow going from adipocytes to the ovary. So we really like to ask very simple questions, because they're easier to answer. They give you clear-cut answers, and you can then ask the next question. So, but why did we pick adipocytes of all those other organs? I think at this school everybody knows the answer, but so the adipocytes are a nutrient-sensing tissue, so that makes sense to look at it in highly nutrient-sensing tissue, so it makes sense to look at it as part of the ovarian response to diet. And adipocytes produce many hormones and other factors with systemic roles in mammals, and that's all true in flies as well. And of course, since there's such important roles that adipocytes play, when something goes wrong with adipocytes, such as in obesity or lipodystrophy, when you have a lack of adipose tissue, they are both linked to infertility in women, and obesity is also linked to various types of cancers and other diseases. So we thought that was a good tissue to start with. And people sometimes wonder where the fat tissue is in flies. So this is a stain with oil red O, which basically everywhere where you see red is where there is lipid stores. So just like in humans, you have fat cells surrounding the major organs, and you have fat cells right under the cuticle, even in the head. And in the case of flies, the adipocytes and the liver-like cells are in close juxtaposition in the abdominal area. So there's adipocytes everywhere here, but in the abdominal area there is a minority of cells called inocytes that are also part of this organ, which altogether is called the fat body in flies. So the question we asked, like I said, we like to ask simple questions, is do fat cells transmit nutritional information to the ovary? In other words, if the whole organism is eating a rich diet, but only the fat cells think the diet is lacking something, can they tell that to the ovary? So we needed a way to specifically manipulate the adipocytes without affecting any other tissue in the fly, so we could do the actual experiment to answer the question. So we took advantage of this system that's called UAS. Gal4 is very commonly used in flies, but the bottom line of the system is that you use a transcriptional factor, which controls expression of genes, and you put this transcriptional factor, which is actually borrowed from yeast, under the control of tissue-specific promoter, and you can use it to control expression of whatever gene you want. In this case, it's just green fluorescent protein, so you can see where genes are going to be expressed, to be able to manipulate tissue specifically. So in this case, this green protein is just expressed using this particular Gal4 driver here, just in adipocytes and no other cell in the body. So using this system, and we also have temporal control over it, so that we only manipulate the expression of genes in the adult fly, so we don't have to worry about developmental problems. So we use this tool to then manipulate the fat cells and ask what happened to the ovary. And the kind of manipulation we did, because of the question we wanted to ask, was if we just, flies have lots of different amino acid transporters, which are required to bring in the amino acids into the cells. So we thought, well, if we want the fat cells to think there's a lack of protein in the diet, we can just decrease the levels of one of these amino acid transporters at a time to cause a very small dip in levels of amino acids in the cells. So it could be like mimicking a situation where you might be eating a little bit less protein. So we knocked down these amino acid transporters in the fat cells using the system I just described and asked what happened in the ovary. And what happens when we look at the number of eggs laid per female per day, we induce the knockdown of the amino acid transporters at day 0, and by day 12, you can see for 3 different amino acid transporters shown here, there is a significant decrease in egg production compared to the controls. And this is true for additional amino acid transporters that we tested. So the question is, why is there a decrease in the number of eggs? What's happening in oogenesis that's leading to this decrease? So we looked at lots of different processes in oogenesis, and one of them was the maintenance of the stem cells in that niche that I described. So the black here or here is the control, and this is showing the average number of germline stem cells starting from time 0 until 10 days after the amino acid transporters were knocked down in the fat cells. And you can see that if you have less amino acid transport in the fat cells, the stem cells decrease in number faster over time than in the control. So there's an increased rate of loss of stem cells. And in addition, the only other thing that we found that was going wrong with these flies, where just the fat cells have lower levels of amino acids, is that compared to the control RNAi, or knockdown, we could see that there would be more of those mature eggs ready to be laid that are being held back in the ovary. So this is also reminiscent of what happens on a poor diet. So the next question was, so if we're knocking down amino acid transport in the fat cells and we are seeing these 2 effects in the ovary, what is the signaling pathway in the fat cells that's involved in the sensing of amino acids? So as I already mentioned earlier, the TOR signaling pathway, it has a very important role in controlling organism homeostasis, especially in response to diet, different kinds of stresses. And as I already mentioned, the insulin signaling pathway can send inputs to the TOR signaling pathway. But this pathway also can be regulated in a more direct way by amino acids. And this is such a well-studied pathway that's involved in so many processes that every time you talk about amino acid sensing, the first pathway that people think of is the TOR pathway. So when we saw that amino acid sensing in the fat cells was affecting the ovary, we thought, well, we should look at the TOR pathway because that's what everybody does, and it's an obvious thing to look at. So we asked, okay, so if you have normal amino acid levels now, and you're not manipulating amino acid transport, but instead you repress the TOR pathway in the fat cells such that you, as far as the TOR pathway is concerned, there is less activity of this pathway involved in amino acid sensing. Can you mimic the same effects that we saw when we knocked down the amino acid transporters? And what we saw, again, we knocked down the levels of TOR signaling, and we asked what happened in the ovary. And we did see, compared to the control, a very drastic accumulation of those stage 14s, this blocking ovulation, which was even stronger than what we saw with the amino acid transport knockdown. So clearly, TOR signaling adipocytes promotes ovulation. So we thought, okay, so TOR signaling is what's mediating the effects of amino acid in adipocytes. But then when we looked at the germline stem cell numbers, we saw that compared to the control, using two different methods to decrease levels of TOR, they were basically the rate of stem cell maintenance over time was basically the same. Like they might start a little bit different, but the rate was the same once you induce the gene. So clearly, the TOR signaling pathway was mediating the effects of amino acid on the control of ovulation of the ovary, but not on the control of stem cell numbers acting adipocytes. So the question was, okay, so if it's not TOR signaling, what is controlling the number of stem cells downstream of amino acid sensing in the fat cells? And so we had to turn to the literature and see what other signaling pathways involved in amino acid sensing would make sense to investigate. And we found this very interesting pathway called the amino acid response pathway. And I should preface this slide by saying this pathway was discovered in yeast, also curiosity-based research. But later, more recently, it turned out that this basic pathway discovered in yeast involved in helping the yeast make the amino acids that are missing in their media. Turns out this pathway operates in the brain of both mammals and of the Drosophila larva, not as cute as the adult, but so the brains in both cases, this pathway gets activated if they have a diet that is imbalanced, is lacking one or more essential amino acids, and then those two different organisms will avoid the food because it's not good for them. So this is acting in the brain. And we thought, well, if it acts in yeast and if it acts in the brain, could it be acting in the fat cells to sense the amino acids? So I'll just give you a brief explanation of this pathway so you can understand how we manipulated this pathway in the flies to ask this question. So the way this pathway works to sense decreased levels of amino acid is that they normally, amino acids need to be brought to the ribosome, the machinery that makes proteins. The amino acids are brought to this machinery by these tRNAs. So first thing that needs to happen for a cell to be able to make proteins is you need the amino acids to be coupled to tRNAs. And usually the amino acids are automatically coupled to tRNAs, and all the tRNAs have a little amino acid they are carrying. But if you have reduction in levels of one type of amino acid, you might start having extra tRNAs that didn't get an amino acid. And then these extra uncharged tRNAs can bind to this protein and activate this protein, which then modifies other factors in the cells and lead to a global reduction of translation. But, a subset of special messages can still get translated to make protein. And in mammals, it drives expression transcription of stress genes in the nervous system is whatever you need to make the animals avoid the food. So the question is, could this pathway then, when we decrease the levels of amino acids in the fat cells, be responsible for that decrease in germline stem cell number? So this is our hypothesis. So we tested it by testing two predictions of this hypothesis, because if this model here is true, one prediction is that if you artificially, genetically increase the levels of uncharged tRNAs, you should induce the same effect. Because normally, you have normal amino acid levels, you have normal coupling, and the amino acid response pathway is off. And if you have reduced amino acid levels, you have uncoupled tRNAs, the pathway is on, and that's our model, that this would cause a decrease in germline stem cell number. If it's really this increase in uncharged tRNAs that's responsible for the decrease in germline stem cell number, then you should be able, under normal amino acid levels, to genetically mutate or knock down the enzyme that's responsible for coupling of amino acids to tRNA, artificially create uncharged tRNAs even when there is normal levels of amino acids. And if this model is true, then this should lead to a decrease in germline stem cell number too. And that's exactly what we saw. We knocked down the enzymes that are responsible for coupling some different types of amino acids to their corresponding tRNAs, and the control here in black is like you should look at the slope of this. There is not a whole lot of stem cell loss. But if you look at, for 3 of these enzymes that we knocked down just in the fat cells to generate extra uncoupled tRNA under normal levels of amino acid, we saw increased rate of stem cell loss. So that was in support of our model. But then another prediction of this model, if that under normal, if you reduce the levels of amino acids by mutating, for example, the transporters for amino acids in the fat cells, and you generate this extra tRNAs, and it acts by activating this protein here called GCN2 to turn on the pathway, then you should be able to have conditions where there is low levels of amino acids, and you have these uncoupled tRNAs. But if you get rid of this enzyme that's needed in the pathway to turn it on, you should reverse the phenotype and get normal levels of stem cells. And that's what we saw. So this is a control. This is if we knock down the amino acid transporters in the fat cells. So now there's reduced levels of amino acids inside the fat cells. But then if at the same time we inactivate a key enzyme in the amino acid response pathway, the number of stem cells does not go down, showing that it's activation of the amino acid response pathway that forces the stem cells to get lost. And so this is a summary of what I just showed, that then the TOR signaling downstream of amino acids controls ovulation, and the activation of this amino acid response pathway controls the germline stem cell numbers. And since we published this study, I won't go over it, but we also showed there's a lot of different metabolic enzymes that are controlled by diet in adipocytes. And it turns out this diet-regulated pathways also have really when you change them in the fat cells, they also lead to specific effects in the ovary. And just to show you how much more complex things are than you think initially, we had shown early on, right, in the first phase of my lab, all the roles that insulin-like peptides play in the ovary by acting directly on the germline. So we just got a paper accepted literally like on Monday, 2 days ago, showing that the insulin-like peptides not only have all these direct effects on the ovary, but they also act in the fat cells to further modify the fat cells, and the fat cells in response to insulin-like peptides can also control several different effects on the ovary. So as you can see, we usually generate more questions than we answer, because now, of course, we figured out some things that are happening in the fat, But now we need to figure out what is leaving the fat cells and going to the ovary to actually affect all of these changes in the ovary. And finally, I would like to acknowledge my past trainees, which have done a lot of the work that I talked and did not talk about, and my current trainees, which are doing a lot of the work that hopefully I will be talking about in the future. And it's really an honor to have been or be associated with all of them, because they're all very enthusiastic. They love fruit flies. They love the questions we are asking, and they're very curious about finding out what the answers are. We also, of course, we're grateful for our sources of funding, and the Drosophila community is incredibly generous. That's because that's been the tradition to share everything ever since the Drosophila was first used as a model over 100 years ago. And finally, I liked the next slide. It doesn't have everyone, so I apologize for everybody from my lab whose picture is not in there. But this was a really nice picture we took at our first lab reunion we had, like not totally planned, but we were at the fly meeting this past April. And it was the first time there were so many current and former members of my lab at the same time in one place. So we all went out for dinner, and it's nice because Leslie and Tian Lu are current trainees. Kate, Alyssa, Elizabeth, and Jessica are past trainees. Some of them overlapped, some of them didn't overlap, but now they all know each other, and they can all have their own little support network. But anyway, so, and I'll take any questions if there's any. Oh, no. I told you not to ask any questions.
Is there a cell in the fly that doesn't express the enzymes?
Let me give you the microphone. So we're recording.
Not that anyone knows of.
Yeah, so, I mean, insulin is really, it's that way as a diabetic. I know the problem of not having insulin. So that really complicates the physiology, because you've got insulin affecting one cell, which is signaling to another, and then
Right.
you have the second cell also being affected by insulin. How do you parse all that out in your brain? You got so many
Oh,
neurons.
in my brain?
Yeah.
Not very well, but what we try to do, but that's why it's so important to be able to manipulate each cell type and tissue specifically. And it's also very interesting because when we first started doing this work, like nobody cared about diet to begin with, but it was also like, oh, of course diet affects everything because every cell needs nutrients. But what you can see is that, yeah, every cell needs nutrients, but there are all kinds of warning signals to help some cells tell other cells what the nutritional status is. And what's interesting, even though not just insulin signaling, but TOR signaling is everywhere, a lot of like hormone receptors are in lots of different places. But what we find is really interesting that even though you have this core pathway in common, there's incredibly specific responses depending on each context. And so you really need to be able to manipulate things separately and try to put the puzzle together.
Daniela, why do you think only certain amino acid has a response?
In the amino acid transporter?
Yeah.
Well, there could be some trivial, boring answers to that. One is like we didn't test every single transporter. There's 40. So the first thing we tested the ones for which there were available RNAi lines. And we basically did not think about the ones that didn't give us any results, because it could be because not all of them are expressed in the fat cells. It could be that the RNAi just didn't knock down properly. But what we did see is that there were 6+ transporters, and they all gave us essentially the same phenotypes. So we don't think it's like a specific amino acid. We think it's like really a general sensing of amino acid levels. Otherwise, we would have seen more specific effects. Actually, there was one transporter that was very specific to one type of amino acid, leucine, that gave us a completely different phenotype. But that will be like, maybe someday will be some other story, but I don't know.
Yeah, I just don't know whether it could be precursor of other metabolites that give you a signal.
You mean?
After the amino acid, how they convert it to other things that they may have shared pathway.
But I have
to
a feeling that downstream of the amino acid response pathway, all those amino acids are converging through common signals, and that's what Alyssa is going to start study in her lab. So I'm not going to study that, but and also you might think it's surprising that just knocking down one amino acid transporter can have an effect. But I think basically it's so important for a multicellular organism to be highly tuned to the dietary, to the nutritional environment, that I think during evolution, all multicellular organisms, there was a lot of evolutionary pressure to develop lots of different ways to downregulate the system. So that it's really easy to recapitulate certain aspects of nutrient deprivation by mutating different pathways, because I think they're all like doing the same job of making sure things are repressed. But if you're on a poor diet and you try to rescue it back to the wild type, it's much harder because any one thing can shut everything down, and that would make sense because you definitely don't want the flies wasting resources, or anybody for that matter. I just care about the flies, but anyways, you don't want anybody wasting resources if there's not enough of it around. Yeah.
Any other questions? Yes, Mike.
It's related to those questions and Anthony's question, but if you decrease uptake of an amino acid into the adipocyte, might that lead to an increase in that amino acid in the lymph or wherever it is in the fly, and that potentially produces a signal that affects other cell types?
That theoretically, it would be possible, but since if you think about it, like the fact that all tissues are taking up amino acids, there are 40 transporters, and our knockdown was about 50% effective in the best cases. It seems unlikely that the effects that we're seeing are because of accumulation of amino acids in the lymph, plus the fact that we were able to manipulate amino acid sensing pathways in the fat body and phenocopy different aspects of the effect. If there is a contribution of that kind, it's really small. But
Any other questions? Yes, over here.
Hi, Daniela. Is it on?
oh, I didn't see you there.
Great job. Love to hear
That's
it.
why I was so relaxed.
I was just wondering, because I'm sure you've thought a lot about what the flies actually eat like in the wild, which is nothing like how we force feed them in the laboratory.
Yeah.
And you probably spent some time on the weekend or at night wondering how all of your findings would translate to the real life scenario where the fly is on the rotting fruit, and we don't have to worry about the more important part of reproduction, which is the sperm. Because she's traveling around with her own package of sperm, so the male is not important here anymore. But tell me how nutrition affects the ovarian stem cells in this other context.
Yeah, well, we don't think about just ovarian stem cells. It's the whole ovary responding. But I think because the flies encounter all kinds of intermediate situations in the wild, that's why there are so many mechanisms and so many ways of organs cross-talking to each other, so that and it's so easy to disturb the pathway because it's highly tuned for any little dip. And we don't think just about nutrition either, because temperature, as Anna is starting to study in the lab, and all kinds of other stresses. Like we all know if we're trying to get an embryo collection, that if you go too often near the flies, or if they're not in the dark, or if you tap on the thing, they're not going to lay that many eggs. So they're responding to all kinds of stimuli and humidity. And in the wild, they will eat something similar to what we eat in the lab in the sense that they will eat yeasts that grow in the rotten food. So I don't think that's such a stretch. Like nobody's home cooking molasses with agar and all that for them in the wild. But the yeast part is definitely in the wild, and they actually respond to that very, very strongly because we've tried in the lab, because since we studied diet, everybody expects us to have, and I don't blame them, but to have like a defined diet where you remove specific nutrients from the diet and see their effects. The problem is like anytime you try to remove this complex food that the yeast is, they respond really badly and they stop laying eggs almost altogether. So, they are just in tune to all kinds of little changes, but.
Great. Well, thank you so much. I, for one, fruit flies have taken on a whole new meaning for me.
Good.
So
I can add more to
I'm
that
sure
if
you
want to
could.
visit my office.
I appreciate them a whole lot more now and what they're teaching us. But thank you so much for being here, and congratulations on all the great work you do.
Thank you.
We very much appreciate it. So join me in thanking Daniela, and then join us for a reception in the gallery. Thank you.